As for the, let's say, gene therapy, defining gene therapy either as a gene addition, which means adding the normal gene to stem cell, or genome editing, which means to modify the DNA in order to increase the production of fetal hemoglobin. As for gene addition, there are data already published. The last one was published by Professor Locatelli in New England last year, and actually the results are quite convincing in transfusion-dependent thalassemia, because after the gene addition, the majority of patients, but 3, became transfusion independent after the infusion. The 3 who were not responding immediately, actually, they responded later. They had a retardation, but now they're responding. So the point with gene therapy in terms of gene addition is that nowadays is not available in Europe. Because although it was approved by EMA in Europe back to 2021 or something like that, then the company withdrew the approval in Europe due to some logistic reasons. It was very difficult to get the approval from region by region across Europe, whereas FDA approved it and is enrolling, although the enrollment is not so easy due to the cost of the procedure. Genome editing is applied either in Sickle and in Thalassemia. The principle is different because the idea is through the techniques of CRISPR-Cas, which is now used for many different approaches. With this approach, the target is BCL11A, which is a suppressor of, physiologically speaking, is a suppressor of fetal hemoglobin production. So targeting BCL11A is possible to reactivate the production or to maintain the production of fetal hemoglobin. And in this way, we know that when fetal hemoglobin is high because it's produced as a hemoglobin either in thalassemia or in sickle, the phenotype expression may improve. Data have been published, both paper in New England at the end of 2024, and the results are very, very promising because for thalassemia, a transfusion-free event is observed after the treatment, and for sickle, the significant reduction of vaso-occlusive crises. So I don't want to say what is better, but definitely genome editing seems to be very, very promising, with less risk, of course, of risk of a viral integration, because we don't use a viral vector for the treatment.